All right. Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. Also with me today is my colleague here at NCCAT, Ed Eng, as well as Vamsi Rayaprolu from the Pacific Northwest Cryo-EM Center, PNCC, and Michael Schmidt from the Stanford SLAC Cryo-EM Center, S2C2. This webinar is a regular event that we host the last Thursday of every month. And along with the exciting biology we hear, all of the talks we host highlight the methods that research groups are using to obtain and interpret the Cryo-EM data that they can collect with the help of National Center resources. And we hope these talks are especially useful for groups just starting to use Cryo-EM techniques. I'll introduce today's speakers, Lorena, Saelices Gomez, and Binh Nguyen, after my colleagues from each of the centers give a brief update on the resources we make available at no cost to the research community. You can save the date now for our next talk on June 30th. Francesca Vallisi will join us from Columbia University Medical Center, presenting her work on the architecture of the human erythrocyte ankyrin 1 complex. And she's going to be focusing her talk particularly on the protein production that allowed that project to be successful. So we hope you can join us again for that talk. Back to today's events, we are recording this talk, and you'll be able to find the recording along with those from our past talks on our YouTube page or by going to the events page on cryoemcenters.org. Logistically, for today's talk, please use the Q&A button to send in questions at any time or upvote other questions you see there. We're going to hold questions for the end of the talk, and I'll be moderating the Q&A session then. So with that, I'm going to turn over to our center representatives. Mike, I'm going to let you take the floor first.
Hi, good afternoon. I represent S2C2, but today I'm live from New York. I've always wanted to say that. We just finished the National Centers meeting where we've celebrated the successes of our users and got together to try and figure out how we can serve them better. At S2C2, Stanford SLAC Cryo-EM Center, we have 3 Crioses and a Talos Arctica, and all of the ancillary equipment and staff to help you collect high-resolution data and train you to become independent researchers in Cryo-EM. There's our website, cryo-em.slac.stanford.edu/S2C2. Visit us and come collect data and learn with us. Bye.
Thanks, Mike. And we can verify you're in New York City because we've got lots of sirens in your background.
True.
All right, next I will turn over to Vamsi.
We're not getting his audio?
Yeah, we're missing your audio again. Not yet.
Can you hear me now?
Yes.
Okay, sorry about that. Hi, I'm Vamsi Rayaprolu. I'm a scientific point of contact, SPOC for short. So just like other centers, PNCC also offers free services to all the users. PNCC currently offers 2 types of proposals. One is a screening access proposal, which is for up to 5 days of on-site sample preparation, optimization, and this is targeted towards low resource institutions. Following that is the general access proposal for single particle analysis and tomography, which gives up to 480 hours a year and is valid for up to 2 years. So approved general access proposals are eligible for training workshops. So PNCC does a bunch of workshops. So if you have a general access proposal, you're approved for those training workshops too. At PNCC, we have a monthly submission deadline for these applications. Each approved proposal will be delegated to a SPOC like myself. who will assist with the data collection, the data analysis, and any of the questions. So I'm actually a microscopist. We have 5 scopes. We have an Arctica with a K3, 4 Cryosis with K3, and Falcon 3 cameras. 3 of our Cryosis have energy filters, and 3 have been upgraded to fringe-free imaging. So for sample preparation, we have a Vitrobot, we have a Leica GP2. Lastly, PNCC is accepting users on site for the microscope operation workshop in the summer. So please keep an eye out on the PNCC website for future workshops, and definitely apply for a proposal today. Thank you.
Thank you, Vamsi. And Ed, you can have the floor.
I'm also here at New York, and we offer instrumentation access on one of our 4 dedicated Cryoses, grid preparation and screening services, including chameleon access, and a cross-training program where you can stay here from a week to a month to even a semester. Talking about workshops, we are offering a workshop at the American Crystallographic Association annual meeting in Portland. That'll be July 29th, workshop 5, Cryo-EM sample preparation training using centimeter badges at PNCC. One thing to note, early registration is May 31st, and if you join ACA, please consider joining a society, either the Cryo-EM SIG or the Service SIG at ACA. So please go to nccat.nysbc.org and find out more.
All right, thank you, Ed. I'm going to stop sharing and introduce today's speakers. So Professor Lorena Saelices Gomez completed her PhD at the University of Seville in Spain, where she worked on the structure and function of glutamine synthetase. She then did postdoctoral research at UCLA and the ETH in Zurich, where she followed her interests in structural biology to study proteins involved in human amyloid diseases. She started her own lab at UT Southwestern in 2020 and uses multiple structural biology techniques, including Cryo-EM, to obtain atomic information of amyloid structures with the aim of designing clinical tools for Alzheimer's disease. And she's already received recognition for the impact of her lab's work, including, but not limited to, an NIH New Innovator Award and a Welch Foundation Research Award. She's joined by a member of her lab, Binh Nguyen, who received his PhD in molecular biology at the University of Otago in New Zealand, where he worked on proteins involved in glioblastoma pathogenesis. After that, he had a lectureship in molecular biology at Vietnam National University School of Medicine. And after that, he moved to UCLA, where he discovered a passion for amyloid research and Cryo-EM that he now continues to pursue in the Saelices lab. So we're thankful to both of you for joining us here today to tell us about your work, and we'll turn it over to you.
Lorena, you are muted.
Okay, do you hear me now?
Yes.
Awesome, great. Do you see my presentation on presentation mode?
Yes, now we do.
Now we do, awesome.
Okay.
Thank you. Well, first of all, I want to thank everyone here and for being here, and the organizers for inviting us to give this talk. We are very excited to show our very recent results. We are going to show results that we literally finished processing a day and a half ago. So I'm really happy and excited to show you this. So the talk of today is about Cryo-EM study of cardiac ATTR fibrils and structure-based design of clinical tools for amyloidosis, ATTR amyloidosis. But before we go into that, into the specifics, I want to give a brief introduction about what an amyloidosis is, or amyloid disease is. So amyloid diseases are those that are associated with the formation or deposition of amyloid fibrils. And these amyloid fibrils deposit in different organs. That depends on the disease. And that deposition leads to organ failure or tissue failure and death. These amyloid fibrils are very specific. These amyloid fibrils have very specific characteristics. First one, they're made of protein, right? And they're elongated assemblies of this protein that it forms these fibrils that are the ones infiltrating in the organs and causing the tissue damage. But the structure of these proteins in the fibrils is very specific. First, the same protein is the one that is piling up, forming these fibrils. And the way that it does it is just by binding in with a beta sheet rich core with a highly interdigitated interfaces of side chains, just like being very close to each other and forming a very compact and strong interface. And that makes these fibrils very resilient to degradation and clearance. They also have this capacity to template aggregation by providing nuclei. So normally an amyloid fibril is made after the protein starts nucleating, kind of like in crystallography, right? So you have this nucleation phase, and then that elongates into fiber formation. But if you have small fibrils, same way that you have with crystals, if you have tiny crystals, those can seed the formation of new fibrils. And that's a process known as seeding. And they can be detected by different methods. And these are important because I'm going to be mentioning them later in the talk. The first one that I'm showing in here is the cross-beta diffraction pattern. So when you align these fibrils and then you inside an X-ray beam through the fibrils, you see this diffraction pattern that is very characteristic of the structure of these assemblies. They can also be visualized by electron microscopy. And because of the structure as well, they have this capacity to bind to different dyes, and you can measure aggregation of a protein just by following the fluorescence of these dyes that come up from the binding to the fibrils. The thing is that there are a number of amyloid proteins or proteins that are involved in this amyloid formation. The one that I'm going to talk to you about, not the only one that we care about, but the one that I'm going to talk to you about is transthyretin, also in short TTR in here. So this protein deposits in different organs of the body and causes ATTR amyloidosis. This disease was thought to be rare, but in reality, that's not the case. 4% of African Americans in the US and West Africans carry a mutation in the gene that put them at risk of heart failure because of this disease. And also 25% of us, when we reach 80 years old, are going to have TTR deposits in our hearts. And some of us are going to die of a heart failure or heart attack, and we are never going to know that it was coming from this. The process in which this protein aggregates is the following. Transthyretin is a functional protein, okay? So this protein has a function. The function is the transportation of thyroxine, which is a hormone, and also retinol or vitamin A through the binding to another protein, which is retinol binding protein. So it's a transporter in the blood. And normally is in the form of a tetramer. This is what you see here. So you see like a dimer on the left and dimer on the right, and this is the hydrophobic pocket that binds to thyroxine, which is the hormone that transports. When you have a mutation in the gene of transthyretin, or because of a gene, this protein dissociates into monomers and starts unfolding, and that leads to nucleation and polymerization. Now, the accumulation of these fibrils happens outside of the cell. It's an extracellular deposition, but it's systemic. That means that this protein is mainly synthesized in the liver, is in circulation in the body, and then it deposits in pretty much everywhere in the body that you can think of. And the clinical presentation varies a lot, patient to patient. Like in here, I'm giving you an overview of different symptoms that you can find in these patients. Not every patient has all the symptoms. Not all the symptoms are in every patient. So this is just very variable. The main two clinical presentations are neuropathy or polyneuropathy and cardiomyopathy. And the same patient can have either or both of them. So good for the patients. There are a number of treatments available for these people. These three are the ones that are already approved by the FDA here in the US. So the first one that was ever used for the treatment of this disease was liver transplantation. And liver transplantation, what it does is to replace the mutant form of transthyretin for a wild-type form. And that back in the days, before even we knew that the wild-type form of transthyretin can actually aggregate as well, which is these 25% of us. Before they even knew that, it was thought that the wild-type form was more stable than the mutant, and that's exactly what happened in most of the cases when you do the liver transplantation, and that would slow down progression of disease. Now, we also have gene silencers for these patients, and what they do is just decrease the expression of transthyretin, so there's less transthyretin in circulation, and therefore less aggregation. And then there's another type of treatment, which is stabilizer. So what they do is to bind in the middle of the tetramer, so those are called tetramer stabilizers, and they would shift the equilibrium towards the tetramer formation, and therefore delayed aggregation. But the problem about these treatments is that they target the functional form of transthyretin, and they seem to have very limited effect at late stages of disease. They work beautifully when you treat the patients really early, but the problem is that these patients, because of how rare this disease is or how poor the diagnostic tools we have, because of that reason, they're often diagnosed very late. So by then, it's already late for the patients to be treated. So a big challenge in this disease is to find a treatment for late stages of ATTR amyloidosis. The second big question of this disease, or the question that we are all wondering, is how do you explain this phenotypic variability, right? It's huge, and there's no pattern, right? There's certain mutations that are more associated to certain things, but it's not even clear. Even within the same family, you can have a lot of variation in terms of onset, symptoms, etc. So what is driving this? And I'm going to start talking about this. So because we are structural biologists, we relate everything to structures, right? And we have reasons to do so. But the first answer, or the first hypothesis that we had, was like, well, it might be driven by structures, right? So maybe the fibrils are formed in certain patients, have different morphology or different whatever, and then that would lead to one symptom or the other, right? And we have reasons to believe that that's true based on previous studies on tau protein, which is associated with Alzheimer's disease and other diseases called tauopathies. Well, the Shares lab at MRC in Cambridge, UK, they found that there is a number of structures of tau that this protein can form, and the structure is linked to a specific disease. In other words, each disease is associated with a particular fault. And if you test different patients within the same disease, for example, here I'm showing you Alzheimer's, every patient of Alzheimer's has the same faults. They can have different morphology of the filaments, like the filaments can bind in different ways, but the fault of each filament is the same. So we thought that maybe this is the case for transthyretin. So the question that we are trying to answer, and what we are going to talk about today, is whether this is what we find in ATTR patients. Do we see the same fault in all the fibrils extracted from cardiac amyloidosis from the heart of these patients? So in other words, if we take fibers from the heart of many, many different patients, how do they look like? Do they look the same as we can see here in Alzheimer's, or maybe they have different morphologies? So to answer that question, we just pretty much got a bunch of heart tissues and extracted the fibers from those. They have different symptomatologies in terms of neuropathy, but they all had cardiomyopathy, so they were all cardiac ATTR patients. We have both males and females, different ages, and they are coming from autopsies or from the heart that was transplanted during heart transplantation. So we extracted this tissues using the method developed by Schmidt et al. and described in 2019 in Nature Communications. And then we determined the structures of this using cryo-electron microscopy. But I'm not going to talk about this. My cryo cowboy will. So here's Binh. Binh, please go ahead.
I hope you guys can hear me. Hello.
Yes.
Can you hear me?
Yes, we can hear you. Thanks.
Yeah, thanks. Thanks, Lorena, for the introduction. My job is pretty straightforward. So to determine as much structures as I can from whatever the patient sample that we received from our collaborator. So the first one that I'm presenting here is the wild type, ATTR-wild-type. So can you go to the next slide, please? So if you see so the image on the right is the fiber length, right? And it's in a zooming section of it is showing the layers stacking on each other, and it's along the z-axis of the fibers. Now, if you turn, well, all these fibers are single protofilament, and so that using just C1 symmetry, and it's very similar to the one that's published in, by Smith. So if you turn that fiber 90 degrees towards you, you end up having the map just like so on the right, on the left, top left. And the fiber have two fragments. The N-terminal fragment ranging from Pro11 to Lys35. And this fragment is a one classic case of the steric zipper, where all of the hydrophobic side chains hydrophobic side chain, the side chains of hydrophobic residue are pointing toward each other to form a steric zipper. The C terminal is more interesting, where the first 30 amino acid, which is ranging from glycine 57 until around isoleucine 84, is forming a hydrophilic core. But then the rest of it is also as a normal steric zipper. Now, what we also found is that, can you go to the next slide, please? What we also found is that consistent with the previous finding from Smith is that we find 3 unidentified density as showing in the black arrows there. We don't know what they are, but we think that the 2 arrows on the right may be potentially associated with the calcium, because we extracted the fiber from the heart. But again, we use a lot of EDTA during our sample extraction, and also the zipper in there is hydrophobic, so we don't know, to be honest. And then we also run the other 2 small analyses. Just the first one is the residue composition that contribute to the fibers. So what you have there is the yellows, the residue shown in yellow is the hydrophobic. The one in green is the polar residue, and then the acid and the base is the red and the blue. And in there you can see three area that contain the hydrophobic residues. And with these, and then we did the second analysis, which is using the energy contribution of it, and just to see how well are these contribute to the how well are these hydrophobic residue contribute to the stabilizations of the structures. And as you can see here, the one showing red is when the one that has the lowest energy contribution compared to the one that showed in the blue. So what if you can imagine like the energy landscape of a protein folding, right? The lower the energy, the more red is showing here. So the 3 areas that showing red we thought to be the main core of the structure, contribute to the main core of the structures, right? And then here is the very scientifically serious debate in our lab whether or not the shape of our fiber looks like. So one theory is saying that the shape of fiber looks like a heart, and as you can see, the blue vein is sort of corresponding to the blue residue in there. And then the second theory is that if you tilt your head 45 degrees to the left, it looks like a strawberry. But yeah, if you can go to the next slides. So we start solving the other a bunch more of fiber, and they all showing the same folds compared to the first one, the wild type one, and it's also very consistent with the one that's published by Smith and colleagues. It's very similar fold. Lucky for us, when we solve the next 2, there are some differences. And it potentially could be based on the it could be because of the mutation that these variants have, and the mutation at position number 84. So if you the glycine 57 in the previous structures kind of forming a joint with these 84S. So by mutation of these 84S, it potentially, open up the hydrophilic cavity. And also at the while we're doing this, there's one published structures using the variants V30M extracted from the vitreous eyes. And they also show a very similar core structures, but the only difference is from the glycine 67 backward to the end terminal of it. So that's different. So now if we superimpose all these structures, these last three structures together, as you see that in that structure there, then you can see that the main core of the structures are very much similar. But the only difference is, again, is from the glycine 67 move towards backward to the end terminal. And we think that that is one possibility of contributing to the structural variation of these of the ATTR sample. Another point is that, which is very classic, is the mutation specific causing this variation. And as we see in the I84S, the mutation there somewhat destabilize the regions, and hence, the fragment that closing up on the hydrophilic pocket sort of open up. And if you can go to the next slides. So this is our observation. It's by no means conclusion or anything. It's just observation based on the local resolution maps that we obtain, right? So if you probably the next slide too. So the top left one is the wild type, where you can see a nice uniform resolution across the entire structure. A range here would achieve with the resolution from 2.9 until 3.5. But if you look at the P24S variants, the mutation occur at the position number 24, which is right, the white arrow there, and it's somewhat causing the a lower density or a lower resolution on that end. And then again, with the I84S, the mutation at that tip somewhat destabilize the tip of the lower right corner of that structures. In the case of T68, because the threonine in the normal structure, the threonine is pointing outward into the environment, so the mutation into alanine and that threonine doesn't contribute much to the core of the fiber. So potentially the mutation into alanine doesn't affect the structure at all. So can you go to the next slide? So those are the two hypotheses that we think that potentially can causes the variations in the structures, either or and both of them combined. Yeah, and that's my part. Do you want to continue from there?
Yes, thank you, Binh. So yeah, as he was saying, so this is an interesting finding because we see this structural heterogeneity in ATTR fibrils that is not yet clear whether they are related to any sort of clinical presentation at all, because first, we only have like a few structures, and second, we have limited amount of information about the clinical presentation of these patients. But we are planning on getting a clear idea of what's going on, and whether this structural variation has anything to do with clinical presentation. But so far, from the local resolution, as Binh was saying, it seems like the mutation might be affecting the resolution or the structure of the fibril to an extent. But again, this is not a conclusion yet, it's just an observation, all right? So this is the first part of the talk, and then I'm going to talk about the second part of the talk, which is about getting a treatment for late stages of disease. And they are both related because at the end of the day, again, we're structural biologists. So what we want to do is to do crystallography and structural biology and cryo-EM, right? So we got to find a excuse. And we found a very good excuse. We said, okay, because these lines of treatment, they target the functional form of transthyretin, we believe that might be related to the limited effect that they might have at late stages of the disease. So why don't we just target the fibrils, right? And then maybe in the future we can use different combinatorial treatments to treat these patients at the same time. So in order to do that, we need to use structural information, or we could use the structural information of the fibrils, right? So in a way, we just use the structural information of these fibrils to get what's called structure-based inhibitors of polymerization. And the reason why we did this was because I joined the lab of David Eisenberg at UCLA, and a year before they published this beautiful paper. And they found, or they showed, that they can design peptides that can cap the tip of the fibril and stop polymerization of a fibril just by knowing the structure of a very small peptide of the fibril, right? So what they did, this was done on tau, and this was the paper, was published in 2011. So the first thing that they did was to identify the part of tau that was forming these static zippers, right? And they found that, I mean, it's known that it was bequivec or bequink, and then they took just the peptide of bequivec and using microcrystallography, they solved the structure of those peptides forming amyloid fibrils, right? These highly interdigitated structures that we were mentioning before. So these static zippers, now you can design a peptide that can bind on top of it, and then at the end you just test whether it works. So we wanted to use this, or we wanted to just follow the same pipeline for the design of anti or structure-based inhibitors for transthyretin, right? The problem is that recombinant transthyretin do not or does not form amyloid fibrils in vitro. You can make it aggregate, you can make aggregates with transthyretin, but they're not amyloid-like. They do not bind congoret, they do not bind thioflavin T, they have a very weak cross-beta diffraction pattern. So by definition, I wouldn't call them amyloid fibrils, I would call them just aggregates, right? So that was our first challenge. Our first challenge is to get transthyretin fibrils in vitro. And then after that, it's all kind of like straightforward, right? So we just need to identify the segments, get the structures of them, design the inhibitor, and test them. So that's exactly what I'm going to show you today. So to get fibrils of transthyretin in vitro, we turn to one of the characteristics of amyloid fibrils that I mentioned before, which is seeding. So we know that recombinant transthyretin doesn't form fibrils in vitro by itself. What if we add fibrils to the test tube, right? But the question is, how do we get fibrils in the first place if recombinant transthyretin doesn't form fibrils? Well, we have to go to the source, right? So we went to the heart, we got the fibrils from the heart, and we added to recombinant protein, to recombinant transthyretin. And what we found was that as soon as you add the seeds, like really at this, like pretty much a minute after, you see this rapid formation of amyloid fibrils measured by thioflavin T. So that was a great start, right? So now the next step is to see, or to identify, the regions of transthyretin that are driving this aggregation, right? Or they are most important for aggregation. So in order to do that, the first thing that we did was to just go to the sequence. All right. So this is the sequence of transthyretin. It has 127 residues. And what you see down here is a prediction graph. So this graph is telling you how prone these segments of 6 residue segments are to form fibrils, static zippers. So every red bar that you see here is just a 6 residue peptide or 6 residue segment that is predicted to form amyloid fibrils. So as you can see, it was a little discouraging because it's like, well, pretty much the entire sequence has this awesome amyloidogenic segments. So the first thing that we did was to test whether these predictions were actually true. So we synthesized all these peptides and we put them in test tubes and saw whether they form fibrils. Like maybe if we are lucky, maybe only 1 or 2 form fibrils, but guess what? Pretty much all of them did. They had this cross beta diffraction pattern. You can see, like the beautiful fibrils on EM. So bummer, right? So we are kind of like at the same square zero, right? So the next thing that we tried was, okay, so let's work on the full length protein. So what we did was to mutate pretty much every of these segments that we found to be forming fibrils in vitro. we mutated them to proline or arginine or some bulky residues like tryptophans that could disrupt fiber formation. They're potentially disruptive of fiber formation. So when we did that, we found that only 2 segments of transthyretin seem to be inhibiting seeding and inhibiting aggregation of transthyretin in vitro. And these 2 are strands F and H. You know, they name the different strands of the functional protein with letters, and those are the 2 F and H that we found to be important for aggregation. And it makes a lot of sense that these 2 are the ones, because if you see the tetramer of transthyretin, these strands are the only ones that are completely hidden within the tetramer and within the dimer. Only if you break the dimer into monomers, only if you break the tetramer into monomers, those strands are exposed. And that's what one would think that evolution wanted to be, right? So, all right, we have this information. So now we just have to determine the structures. But standard crystallography requires the formation of a very big crystal that has that size, and then you use an X-ray beam that is quite thick as well, and then you solve the structure using this information. But the problem is that these peptides do not form standard crystals. They form very thin needles. So we had to use a very specific technique that was developed by the Eisenberg lab, that is known as microcrystallography or X-ray microcrystallography. So what you do with this technique is just you fish the crystal and you actually mount them dry, and you use a very small X-ray beam. And of course the crystal is going to decay, but you have to move the beam, you know, along the needle to get multiple data sets, and then you merge them together, and that's how you get the structure. Is a little challenging, but it's a lot of fun. So using this microcrystallography technique, we solve the structure of a number of peptides, not only strand F and H, but also a few more. And back in the days, we didn't have Cryo-EM to see whether those were relevant or not to the actual structure, but now we do. So if you actually compare the structure of the Cryo-EM structure of a fibril, this is from wild type, with these peptides, what you see is that strand F and H are very close to the structure. Also, this one, VABHVF. The other 3 or 4 that we solved, they are not as close, but those are very, very close. The RMSD of those is very low, within like pretty much error of the measurement. So, which is very encouraging, but we didn't know that back then. We just went with the flow. So using these peptide structures, now we have to design the inhibitors, right? And we did this for every strand H and strand F. So we have 2 inhibitors, but I'm going to show you just one, all right? Or the pipeline of this, or of peptide design for just one, okay? So this is the structure of the fibril. You have the side view and you have the top view. This is just a short peptide, right? This is AAVVFT, which is strand F. Then to design a peptide inhibitor, you start by just getting a complementary sequence, which in reality is just the exact same peptide. And then you modify this exact same peptide by adding tags. And those tags, what they're going to do is to increase solubility, and also they are going to decrease self-association. Because again, at the end of the day, you're playing with segments that are amyloidogenic. So if you leave these inhibitors in solution, they're just going to bind to each other, and that's not going to do the job. But if you have the tags, that would increase solubility of the peptide and decrease self-association. Now you have to play with the side chains. You have to modify them. You can also use non-natural amino acids. And the whole idea is to increase the interface, or increase the affinity of the interface, right? And you also can play with the length of the peptide. There's no order for this. You can go back and forth. The whole point of this is to generate a number of peptides. This is all rational design, by the way. And now you use computational design to screen them. So you screen all the things that you rationally design to see which ones are the ones that are the best, or seem to be the best computationally. And now the final ones, you just test them in vitro. Now, I'm going to show you the test, but I'm not going to show you the screening. So I'm just going to give you the best of the inhibitors that we had. That again is a cocktail of 2 peptides, one that it binds to F and one that it binds to H. All right. And the test that we went to first was just to see whether they can actually inhibit amyloid seeding. So again, we extracted the fibrils from patients and we added to wild-type TTR. And with no inhibitor, you see this rapid formation of fibrils. But with the inhibitor that binds F, you see a decrease of signal. With the inhibitor that binds to H, you see a delay in the seeding and decrease of signal. And when you combine them both at the same final concentration, you have this synergy effect or synergistic effect, and they inhibit completely seeding of these patient sample. Now, again, this is just one patient. We tested these peptides in multiple patients, and we found that at the highest concentration, these inhibitors could inhibit seeding in all of them, and in a dose-dependent manner. So, interesting enough, when we were doing this assay, we were wondering what would happen if we add tetramer stabilizers. And this is a little bit off topic, but I just want to show you this because it's relevant for the story. Because the thing is that we want something that is specific of fibrils, right? We are designing something that is different from stabilization. And this is actually the ultimate test or the ultimate evidence that that's the case. Because we added these tetramer stabilizers to wild-type transthyretin, and we measure seeding of those. And these concentrations that we are adding in here, we knew that were enough to inhibit aggregation of the protein without seeds, all right? So this is without seeding. The aggregation of the protein is fully inhibited at these concentrations, right? But when we added the seeds, they did not stop seeding. And we tested this with all the patients, and that's exactly what we saw. So it's that was encouraging in a way, because that tells us that it's the mechanism of action of our peptides is different from stabilization, and that we can differentiate from them from those drugs in the future, which is very encouraging. Now, we had a hard time trying to explain why this happened, because what we knew back in the days is that if you have a tetramer, that has to dissociate into monomers, and then you have a tetramer dissociation barrier, right? Energy barrier that would need to nucleate, and you also have the nucleation energy barrier, and that would elongate into fibrils, which is the thermodynamically most stable form, right? If you have a ligand that binds to the tetramer, and that would stabilize the tetramer, you also have the ligand dissociation energy barrier, but at the end of the day, we would expect to see at least a delay on seeding. But how do we explain the lack of delay? So I think the way that we explain it, or that we can explain it, is that, first of all, when you have the seeds, you're just bypassing this nucleation energy barrier. And at the end of the day, the fibrils are the most thermodynamically stable form in the whole picture. So you expect to see fibrils regardless of whether you have ligands or not. But that doesn't still, explain why you don't have any delay. I think the way to explain it is just by going to kinetics, because at the end of the day, this is an equilibrium, right? And it can be faster, slower, but you're always going to have monomers in the picture. But the moment you get into the fibril, that's an irreversible pathway, at least in vitro, right? So under our conditions, we believe that this is what's going on. It's just, it doesn't matter how fast or how slow this goes, you're always going to have monomers that are going to be, taken up into the fibrils with seeding. And finally, going back to the inhibitor story, we tested them in Drosophila models. So these flies express human transthyretin in their brains, and that makes them very sloppy, and they do not fly well. They do not, the mobility of them are really, really impaired. So what we measure in these flies is the ability for them to climb. Is a climbing assay. So you tap the flies at the bottom of the vial, and then you let them climb, and then you see how they do. Now we treated them with peptides in the food. That means that we have to change the food every so often, every 2-3 days. And the peptide has to make it to the brain through the gut, right, to see any effect. So this is what we saw. So I'm going to show you a 2D projection of the pathway of these flies, right? Now you do this, and then you record this using a camera, and you can measure all sorts of things, but this is the most visual way to see it, and I think it's the easiest. So in summary, when you treat them with vehicle, they do poorly. They do not move from the bottom, not much. When you treat them with the first generation of our inhibitors, then they start moving a little more. But when you treat them with the second generation of our inhibitors, the combination of the two F and H, then they pretty much behave as healthy, which was very encouraging. And that was also associated with a reduction of TTR deposition in the brain of these animals. That was measured by Western blot and quantification. And this was very encouraging as well. So going back to the kind of like the full idea of why we are doing this, again, these treatments target the functional form. They have very limited effects at late stages. And we believe that the reason why they have limited effects at late stages is because probably at late stages, the main or one of the main driving forces of disease progression is seeding, because these patients are full of fibrils when they are at late stages of the disease. So during that time, probably the driving force of polymerization is seeding. So that's what we believe. And we are proposing to target seeding using a structure-based anti-seeding inhibitors. So now we still don't know whether the structural heterogeneity of fibrils is going to answer the question about what's going on phenotypically in these patients. But we know that the more we know about structures, the more we can do in terms of drug discovery for these patients. So what we are currently doing is to use the structures of the information that we are getting from Cryo-EM structures and translate them into new inhibitors of seeding. And with that, I just want to thank everyone who contributed to these amazing projects, especially my lab. I love them, really. They are fantastic scientists. It's great to work with them. I'm really grateful to have them. And of course, everyone is picking particles because I don't know if you guys know or not, maybe not all of you, but for Cryo-EM helical reconstruction of amyloid fibrils, we have to pick the particles manually one by one for 10,000 images per data set, which is a lot of fun. So we are all picking particles left and right. So thank to you guys, because that's the limiting the bottleneck of this project. And of course, I have to thank everyone in the Center for Alzheimer's and Neurodegenerative Diseases, which are fantastic collaborators and peers. The Amyloidosis Center at UT Southwestern, which are also fantastic professionals. The laboratory of David Eisenberg and all the people who contributed to the project at the beginning, the UCLA team and the X-ray team as well, that they were the developers of microcrystallography. And of course, all the Cryo-EM facilities and EM facilities, I can't thank you guys enough. Because you are fantastic. Without you, we had nothing to show. And the donors and their families as well, of course, because that was a very generous contribution, and that's helping us advance in the field. And of course, collaborations. The main one that I want to mention today is the late Meryl Benson, Dr. Meryl Benson, and Barbara Klug-Beckerman. They are the ones providing all the tissues from patients. We have other collaborations that are also giving us some tissues, but the ones that I'm showing you today, are from the Indiana University. And of course, all the technical support that we get from UT Southwestern and other places, and the funding, previous and current funding. And I don't know if Binh wants to give some acknowledgments as well. I'm sure he does.
Well, I'm going to add the ANCAT Center, providing the training for me, the cryo training for me, and I totally recommend that course. It helped me clear a lot of doubts in my head about cryo, because again, we are just like a year old into this stuff. With the help, it's certainly help us moving forward with the cryo. So yeah. Thank you so much.
Thank you both. That was absolutely wonderful. I think there's a large round of applause that you that you can't hear through Zoom. But thank you so much. And it's really sort of astonishing to me to hear you're just starting out when you see so many high resolution structures that you've been able to solve so quickly. That's really exciting and something we really enjoy seeing as well. So please send questions in the Q&A. We have a couple to get started. So actually Binh addressed one in the Q&A box already, but I'll ask it again so that you can answer out loud. But there was a question about doing classification and focused refinement to look for conformational heterogeneity in your different samples.
Yeah. So my answer is that we have not. In fact, we have not. Well, we haven't tried this technique, and so we would love to know how would you approach this. And my email address is down there, and we would love to to collaborate with you for further. But what we did was we solved the entire structures, and then we sort of compared them one by one just to see how different they are, the map and also the model. So that's all that we did so far. Yeah.
And actually a follow-up question related to that. How similar is your processing pipeline for each sample? Do you have like a standard workflow that then works for everything once you've worked it out, or do you have to take each data set as its own?
We're trying to keep everything as consistent as possible. So the way that we do it is we use very similar box size, similar people picking particles too. So it's the same pattern, people picking particle, the same parameters for 2D classification mostly. In 3D, we use we also use the feature list cylinders to start as our initial model, and then we cranked up the parameter from there just to so that's the standard approaching our lab. We have tried also using the initial model from the structure that we have just to see if we can improve the resolution. It does improve a little bit, but then again, we just want all the structures to be comparable with each other. So that's how we do.
The problem about using a model that is already built as a reference for 3D classification is that you're biasing what you're seeing in a way. So that's why we're starting from the cylinder. So we have a clear kind of like classification of all the things that we have, especially because we have variations that are very subtle. These are not, we don't call them polymorphs, right? We call them just like structure perturbations or structure variations, because it's just pretty much the same conformation. It's just like it varies slightly depending on the mutation or whatever.
Thank you. There's another related cryo-EM question before I pivot over to some more biology related questions, but there's a question about the interpretation of local resolution, and how would you interpret some of the low local resolution spots that are far away from the mutation, and would that give you any idea on inhibitor design?
Yeah, like take it with a grain of salt. Like I said, it's just what our observation that we see. We're not jumping to any sort of conclusion yet, because it's the local resolution. There's a lot of factor contributing to the local resolution, and it's not just the fiber itself. It's the process of carrying it out too. So, but it's the observation that we see, and we just put it out there.
The way that we make peace with it is that if you see some perturbations in the local resolution at the outside, but it's subtle and it's kind of like overall, I would think that might be related to the sample, right? Like maybe the resolution is just not that great on those regions because of just it's less order because it's on the outside, or maybe the sample has, or I don't know, even the collection or some other stuff. But when you see, I mean, for us, it's very intriguing to see that those the main perturbations that we have in local resolution are close or near the mutation site. That would makes us wondering whether the mutation is leading to disorder in the structure, and that translates into lower local resolution. But again, we have only a handful of structures, so there's nothing to conclude here. We just need to solve, many more and see the same trend or something, or maybe do some other studies to confirm or rule out this idea. And as for using that information for the inhibitor design, absolutely. Right now, the thing is that we only have like a handful of structures, but eventually our idea is to have enough structures so we can have an idea of where to tackle the fiber formation. So far, the structures that we have very intact strands F and H, which is good for our inhibitor design because those are the ones that we designed. So all of the structures that we have so far have an intact strand F and H. So, but we don't know if that's going to be the case in future patients.
Thank you. I'm going to go next to a question about your structures. Lin Chen asked if you seed the wild type TTR with the mutant that has a different amyloid structure, do you get a wild type amyloid structure that now looks like the mutant, or those experiments you've been able to do?
Say again. So if we have what?
If you seed the wild type TTR with a mutant with a different structure.
Yeah. The thing is that so far it's not clear whether we can reproduce truthfully the structure that we have from the patient into the seeded fibril, and that's another thing that we are working on. But probably it will take some back and forth because the whole idea is to be able to seed the exact structure, right? But being in vitro without any cofactors or any environment, it's going to be challenging to reproduce the exact conformation. So having said that, in what we know from patients is that after liver transplantation, you replace the mutant form with a wild type, right? So that's what you do in the liver transplantation. And now the circulating transthyretin is wild type. We know that this wild type can incorporate into the fibrils in the heart. And that's another thing that we don't know about these patients. We don't know if they had a liver transplantation done. We don't know how much wild type they have in the fibrils, which is something that we can calculate, but it's an assay to do. How much wild type they have in the fibrils, because that might interfere with the perturbation that we see from the mutation, right? But the answer to that question is I would expect the wild type to incorporate into the fibril as a wild type. Like, for example, let me go back to this. Well, it's happening here. Sorry. But here, right? Here. So I believe that probably this mutation I84S, which is closer to the wild type, I mean, our hypothesis, and that's something that we are trying to figure out, is maybe this closer look comes from the fact that this patient has more wild type in the fibril. So some of the layers, most of the layers might be wild type. And if that's the case, the wild type would be able to close the channel. I don't know if that answers the question.
Yeah, I think that definitely addresses the question.
Okay.
And I was gonna say, we're coming up on the hour, so probably a number of people have to pop off to their next meeting. So thank you everyone for joining us today. We did not quite get to all of the questions. If Lorena and Binh, if you can stick around for one more, I'll toss one more question at you
Sure. Yeah.
But thank you everyone for joining us. The national centers are here to support your science. And so please let us know how we can help you. And hopefully we see you again at next month's webinar. All right, and I'll toss in one last question, and I'm going to be a little bit biased since we focus on the cryo-EM side, but there's a question about your processing. When you start with a featureless cylinder as your initial model, how do you overcome local maxima in twist and rise parameters to arrive at the final parameters that give you your high resolution reconstructions?
Yeah, so in our pipeline, when we start with the featureless cylinder, we do a we start with that, and we low filter at low pass filter at a very at 60 I think it's 60 angstrom, and then all the other parameters that we have are very vague, like the angular is sorry, the heel pitch is about 7.5, and we keep tau as low as possible. We have a wide angular search too, and we just keep it running it slowly to build it to build it up and to get to the density, the map density that we see. It's a lot of judgment, or you see and you judge, and then whether or not you should move to the next step, or should you stay on that step and run further with the more broader parameters that you can run, right? And then once and for the twist and the rise, the rise is I started 4.75. For the twist, I measure using the 2D. So I either run a bigger box size in the 2D to see the twist, to see the crossover length, or I stitch them, stitch the image together just to achieve the, and that's where I started. And then when it gets to around 5 or 5 to 6 angstrom, I crank it up the parameter a bit further and slowly doing it. And when it gets to around 4 point something or 5-ish, I take the whole thing. I take the whole thing and do the auto refine, and that's where after the second round, that's the first round auto refine. The second round auto refine, that's where I start to optimizing the twist and the rise. But yeah, that's the overall pipeline that we use. I would love to share with you more if you if you have any more question.
Thank you, Binh. That is incredibly helpful, the level of detail you're sharing. And I think I did notice you shared your email in the Q&A. So hopefully you make some connections through there. And I noticed, Lorena, you're furiously answering away some of the last questions. So if you don't mind, what I can do is pass the unanswered questions to you. And if you wanted to send me answers, I can post that along with the recording, and we'll make that available to everyone. I'll send out an email when we have that material posted.
Yeah, totally.
Okay, fantastic. So we'll go ahead and wrap up. Again, thanks everyone for joining us. And we look forward to seeing you again.
Thank you.
Thank you. Thank you for having us.